All right, so let's get started today's class. So we continue electrophysiology. So today we want to compare different categories of electrophysiological recording techniques, which research questions each technique can best address. So to the point of our class, the core is you have to have a question, okay? So what kind of research questions do you have? And then you can fit each technique, what techniques would be necessary to resolve your question. In some cases, you may have to invent new technique. That's a good example is how we measure action potential based on voltage clamp method, which actually led to Nobel Prize of describing action potential in a very systematic and quantitative manner. Okay, and then we will talk about different types of tissue preparations in electrophysiology experiment. In reality, tissue prep is like almost the most important. So that's where you put a lot of effort in your skill, your thinking, all your experimental techniques. And prep is so important. That's why in this class, we try to give you hands-on experience. Okay. So techniques, we already talked about common electrophysiological approaches and common electrophysiological preparations. So I just have to say that we have to cover a little bit much. So I may be a little fast, so bear with me and let's keep focus. All right. So let's see. Sorry. Okay. So let's talk about extracellular and intracellular recording with electrodes. So first EEG, this electroencephalograph is putting an electrode on top of your scalp. So that's a skin through bone, and let's say there are neurons of interest. So this is how we measure with the EEG. And there's another technique called the ECOG. So that electrocorticography, this is when you remove skin and bone in a surgical setting. Let's say, for example, I have a patient with a epilepsy, and it's important for neurosurgeon to identify where are the focus of epilepsy so that we can get in and cut it out to remove and treat epileptic patients. So in that case, you have to have this surgery procedure, and then you put this electrode on top of the brain parenchyma, which is of course piameter, and then you measure. So you have to, this removing skin and bone, electrical conductivity is way better so that you can have a better signal for this. And then FP stands for field potential. And that now you put into an electrode into the actual tissue, and then nearby neuron, you can measure unit spikes. So this unit, single unit means a single neuron. And then even in some cases, you can actually poke into inside neuron. So I want to show about the typical examples of extra and intracellular electrode signal on the oscilloscope. This is a cartoon, but let's think about when we put an electrode extracellular, first intracellularly, we already discussed about action potential. So that's what you put the electrode inside the cell, and then you measure it. So baseline of a minus, let's say 65 millivolt, and it can go to up to 30, 40, and then come back and refractive period and return. So that's what you know about intracellular recording. And extracellular recording is actually the electrode is not inside, but outside. What that means is you see the changes of electric charge polarity, that extracellular, in fact, is not going up. For depolarization, it goes down. So that's a difference when you look at extracellular recording data, and also at the same time point out that the unit of this, you see this is a millivolt range, while extracellular recording, you see it's microvolt. It's almost orders of magnitude, in this case, 1,000 times differences in signal. So you notice for this. So each action potential exhibits a different waveform due to its placement relative to a neuron. And so the shape is really a characteristic of specific neuron. Okay. So extracellular recording, action potential can be detected in extracellular space near the membrane of an active neuron by measuring the potential differences between the tip of a recording and ground electrode. And where do we put ground electrode? It's far from extracellular position. And so what we observe is a brief and alternating voltage differences between the recording and ground electrodes. So what a good thing about this extracellular is relatively easy and simple, because you don't need to find out the cell and poke inside precisely. And this is can provide, let's say, Yoo-Sung-Young?
Yes, sir.
Yeah, can you guess what this is?
Local field potential.
Exactly, local field potential. And for this extracellular recording case, what we are measuring is outside sum of all post-synaptic activity within the volume of neural tissue. And this is some synaptic activity of neurons in nearby extracellular recording electrodes. However, there's a limitation. Seung-Young again, what's the limitation of extracellular recording? It's impossible to measure?
Self-selected.
Great, yes. Because it's outside, if it's not like strong spike, like action potential, it's hard to measure. So sub-threshold potential changes such as excitatory postsynaptic potential or inhibitory postsynaptic potentials are difficult to measure. Okay, so I put some of the local field potential recording with microelectrodes. This is an example of real data from a literature. So you can see, so there's a reference electrode and recording electrode, which is nearby extracellular. And you have this OPMs. So this amplifies and gives you an output. So let's take a look at the output data together. So this is raw signal, and you see, usually we present the time versus this voltage changes, differences. And this one is a little complicated. So you have to look at it in like expanded. And the way to look at this data, is if you want to see the overall shape, so that's low pass filtering, that we call local field potential. And we are interested in the spike activity, then you can go through high pass filtering. Here, low and high means a frequency range, okay? So that you can now look at the spiking activity. So how we use this with the microelectrodes, as I said, there is a tetrode or MEA multi-electrode array, and which you will poke and looking nearby this electrode. In this case, you can see there's 4 electrodes nearby, and it can be extended to as a multiple electrodes array as well. So the configuration of these electrodes is we can see what would be limit of if I have only one single electrode. Then with the one single electrode, the number of neurons that causes recorded neural activity as a probably one single signal, then we call this a single unit. Let's say next is Min-sung. Can you guess what this is? A single unit? So
I don't know.
action potential, it's related to.
One.
Exactly. Yes, that's a
Oh,
single.
one.
Yeah. So that single unit spike is really a function of the target neurons. Hey, this is target neuron, their shape, and whether they are big, small, the size, and the distance from the electrode versus this target neuron all affects the shape of the single unit spike. So for that, we have two specialized type of electrodes for multi-unit recording. That means we can have measure from the multiple electrodes. So that's a tetrode. As I said, it's a 4 individual electrodes. And the other is, as I already said, a multi-electrode array, which is grids of individual electrodes or groups of tetrodes that are arranged into a single device. So for MEA, if we have a multiple electrodes, there's a way for us to, some of the electrode, we can use it as a recording, passive recording, but because we have a multiple, some of the electrode we can actually use as a stimulation. We can inject current, okay? And then we can see the response. So for stimulation and extracellular recording from several neighboring sites at one time. And this multiple microelectrode, the number is typically 4 to even over 100. So like state of the art is making this like, let's say 1,000 electrodes. But you can imagine that would be a huge data set and work. But that's how things go. We can go even very complicated and multiple numbers of neurons simultaneously we can measure. So in vitro preparations with a brain slice over a grid of many micro electrodes for extracellular recording. I will get back to this later. And in vivo preparations, we can do, I showed you MEAs, multi-electrode arrays, and tetrodes you can insert into in vivo. So we can put in intact brains of live animals for multi-unit recordings. So once we get the signal, and now this is time to analyze, that we call spike sorting. So as you remember that those a spike signals, and this one, if you expand to see, you will see the various shapes of the neurons, and that we call spike. And that has been detected and arranged. And you see multiple, it looks like there's something there. And so first, with the time, you have to have a nice align. And then from here, you need to catch the features. So then you can actually see there are like 3 different type of basic shapes are existing. So that feature can be extracted, and different classified groups can be regarded as different neurons, and you can color them, and then you can replot them. And what's the output of neural spike sorting? This is an input, and the output is in fact this bottom one. You can see once you can distinguish which one's shape is a blue one, then you can find out blue one action potential at this time point. And then simultaneously, you can find the red one from here, and then you can plot it. So this is a like spot. X-axis is a time, and this is what's quantitatively showing at what space and time a single unit are making a spike or action potentials. So to give you an idea what's happening here is, let's say there's an electrode where nearby, let's say 3 different neurons exist, and they will fire at their own pace or condition, and that will be mixed up here. So this one, you expand, you can consider like this, and you give different shapes, different coloring, and then only this time point you put the dots of this firing diagram. Okay, so it's conceptually not that difficult. So spike sorting is this single unit data from this single unit from recording of multiple electrode at once, and this is the output. So the issue is identifying individual cells based on their spiking activity, and we assign the waveforms to a particular cell. Here the assumption is, of course, each neurons produce a distinct type of shape. You see these, and that's in general true. And how we process it, these signals are processed using a triangulation method based on the position of electrodes and the strength and characteristic shape of a recording. And you see the output is a specific and reproducible waveforms for each individual. In this case, you can see blue, red, and green, 3 neurons shapes are identified. And so spike times for each neuron so is as an output. Okay. So let's get into a little bit deeper into how extracellular recording to this spike trained. And this is a reference, so you don't have to have too much of a know everything, but this gives you a concept. Hey, we put insert an electrode into neural tissue where like nearby neurons and outside far neurons, and let's look at the raw data. And you can see these raw data, and you see this recording and reference, electrode and amplify, and then you have raw data. So as I said, you can see a local field potential by having low pass filtering. So this means that this x-axis means a frequency and y-axis is transmission. So you only filter lower frequency, then you can see overall shapes, and that we call local field potential, right? And then on the other end, you can only filter, look at the higher frequency and removing this local field potential, then there you can do spike train, and then you can do spike sorting. Then for example, in this case, there are an example as a cluster 1, 2, 3, 4. You can have a multiple clusters, and you can actually count them as a number of spikes. And then you can replot here with this raw data, high pass filtering data, and you can now output spike trains. You see this a triangle for different color stands for different neurons, and you can actually have this spike train as an output. So this is really measuring neuronal activities directly in time. So depending on your hypothesis or your stimulation or your perturbation of the system or the animal behavior, specific behavior, you can link to how these specific neuron activity can be correlated and studied. So with that, we can go to connectivity and timing within we can talk about neural network. So the tool is investigate the activity of dozens of neurons at a time. We can also manipulate specific neurons and monitor the effect of other neurons recorded by the array. Okay, you can manipulate, you can stimulate, and you can monitor specific neuronal activity. And simultaneous responses of neural circuit provides information on neuronal interactions. You can have this electrode, not only one, but the multiple, and the other neurons. You can actually find the interactions among and between neurons, that interaction and spatial temporal information about neural networks. So people say about connectivity. So brain connectivity means how neurons and neural networks processes information. So there are different type of connectivity people are discussing. I want to talk about like three representative one. First is structural connectivity. So structure or anatomy. So that is about axonal fiber pathway. So are they structurally connected? So that is anatomical links. And there are other kinds of connectivity we talk, which is a functional connectivity. So this is about statistical dependency in different brain region that we talk about correlation. Let me give you an example. Hey, we watch a brain, let's say it can be fMRI to watch different brain region, and we watch activities of the brain, neural activity, and we somehow we realize, we ask the patient or subject, hey, just do not think anything. In Korean, 멍 때리도록. Do nothing. And then we take fMRI images, and we realize this area and the other area, somehow their activity goes together. So if we find that, and we can have a map of the whole 2D or 3D of the map, hey, somehow different brain regions are coming together. Their activity is somehow correlated. So that is what we talk about functional connectivity. It doesn't necessarily mean this functional connectivity has to be directly linked as a structural connectivity though. And in the end, we ask question about causal interaction, causal, which one is cause and which one is effect. So how information is flowing, and that we call effective connectivity. All right, so let's get started with the intracellular recording first. So there's a cell, we have an electrode, we have to poke into the cell, and then we record. So this is an example picture. If you look at it, this is just fascinating. You see, this is like real digital signal, peak, peak, peak. And this is so amazing if you watch this. And what we detect is small graded changes in local membrane potential caused by, let's say, EGBOM. Can you guess what this is? When we do intracellular recording? Yes. Exactly, yes, synaptic events, okay? And we puncture a neuron or axon with a microelectrode. We are measuring the potential differences between the electrode and the reference electrode, which references has to be positioned outside of the cell. And so intracellular, and I want to talk about a key technology for intracellular is patch clamp techniques. Just give you an idea that Bert Schengman and Erwin Neher got Nobel Prize by developing this. So what is patch clamp? There's a glass pipette, which you can imagine glass pipette. There's a tool with a bunsen burner, like to fire, heat them, and then you pull them, and then fire becomes very, very, very narrow, and then you pluck it. Then you can have a super fine tips as a glass. And this is Ji-hoon well described today, that the diameter is only a micron order, so tiny, okay? And you can have a suction while you try to touch a cell, and then you have a mild suction. And what happened? This is so small that this can have a tight contact between the pipette tip and the membrane. And we call that, so this is a, let's say, Guione. Okay. Yes, exactly. So that's glass micropipette to make tight contact with a tiny area. So we call that as a patch of the neuronal membrane. And all the ions flow with, when a single ion channel opens in the patch membrane, must flow into the pipette. Now the pipette is connected to electrode and you are measuring the current now, okay? So the pipette resulting in electrical,
Current.
exactly, current. And you can have a seal resistance over 1 giga-ohm by cell attached patches, often referred to as a giga. It's difficult, we call it as a giga seal. It's like complete sealing, okay? So that's a cell attached mode. What about whole cell mode? So now you know that you've attached it, okay? And you can have a strong pulse of suction, then you can puncture, make a hole. So what happens? Cell cytoplasm is now connected to inside the glass micropipette, which is filled with a solution here, okay? So that we call whole cell mode. Now you are actually connected. The interior of the pipette becomes, let's say, 최유진. Yeah,
Continuous.
exactly. Yes. Now it's continuous with the cytoplasm of the cell. Now you can measure the electrical potential and current from the entire cell. That's why it's whole cell mode. And this is now, you are actually connected to whole cell or single cells inside. So this now allows diffusion or exchange. What it means, you can put a drug through here and then drug can get inside the cell directly between the pipette and the cytoplasm. And that's a way to inject substances into interior of a patch cell. For example, now, let's say you measure the cell and now you want to mark the cell. What can you do? You can put some dye into it. The dye will diffuse into the cell. So that single cell, what you measured, after your study, you want to know how the cell looks like, okay? You flow through a dye. So later on, when you check that out, you can find one single cell which has the dye. So this is an amazing method to even identify one single cell and you can measure that. So another patch clamp technique is called inside out and outside out. So that sounds a little bit weird, but let me talk to you. We have a cell attached mode, okay? And you already suck it mild enough to having gigaseal. Now what you can do is you actually slowly, gently pull it off. Then what happens? This lipid bilayer can tear apart. You actually can have this single ion channel detach it. So now with this, you can actually study about single ion channel. So inside out, this is called inside out because it's inside, all right? So let's say, 양은아, what would be, is it intra or extracellular surface of a patch is exposed?
Intracellular.
Exactly, you see the inside is exposed, intracellular. The measurement of a single channel current while changing, now what you can do is now you can change the outside medium, whatever in what you want. That's mimicking inside the cell. So now you can change the medium which is exposed to intracellular surface of the membrane so that you can study influence of intracellular or cytoplasm molecules on this ion channel function. So you can think, hey, what about the other way around? I want to change outside of the milieu of the cell and I want to see the functional changes of ion channel, which is possible. So let me show you how. So what you do is, in fact, you retract the pipette, okay? And with the first, you see the difference? Now it's a whole cell mode. You strong pulse of suction make this a tearing of the membrane, and then you actually pull it apart. What happens here? And somehow, actually this happens many times, this teared membrane can re-anneal to make this format. And you see the orientation of the ion channel. Now the outside goes out of this pipette. That is called outside out mode. So it's to expose neurons now extracellular surface to the bath, bath by outside of the pipette. So now you can study how this channel activity is influenced by extracellular chemical signals, such as you can put a neurotransmitter, how things work, how ion channel function change. So is this clear? I mean, this is something, people who have done in their life for patch clamp, somehow they may not have done inside out, only they've done outside out. So I'm talking about the differences and how this is coming from, okay? This is reference, but cell attached recording and mild suction and you pull it off, you can have inside out recording. While you have this tight contact, and then what you do is a strong pulse of suction and then pull it off, then you can actually make outside out. This is really fascinating. And so you can study a single ion channel with a different inside and outside the milieu you change, and you can study the function of the electrical functionality of single ion channel. All right, so now let's compare this pros and cons of different patch configuration. So let's see. So cell attached mode, the good thing is we can study single ion channels. We can record, we can study channel properties, will not change it. And that's physiologically relevant because you didn't kill the cell, you didn't perturb the cell much. And this is one of the easiest patch clamp method. Okay, what are the limits of cell attachment? Exact membrane potential is unknown, and it's difficult to control the environment of single ion channels, as you see, because you attach the cell with a glass pipette. So now you make a puncture and continuous with whole cell mode, and now you have a glass which you can pour whatever you want, okay? So you have an ability to manipulate the cells. Can you guess what that is? Cytoplasm, pharmacologically. And now you are it's not like extracellular recording. It's actually connected directly to the inside of the cell. So the current will be large, okay? So the signal is large, easier to measure and deal with. And what are the problem? Now you puncture the cell, cytoplasm is continuous within the recording electrode, and the cell content may be diluted or leaked out. So the cell physiology might have changed. So you have to be careful in that aspect for whole cell mode. What about excised patch, which is what you just see inside out, outside out? You can have a single ion channel, which is a tiny, tiny, tiny one, and you can record. Recording can be taken from individual ion channel. You see, this is a fascinating. That's why I actually got Nobel Prize, because it allows to study single ion channels. And you can have an excellent environmental control, because once you take it out, if it's inside out, you can actually change the bath condition, which mimicking the cells inside, okay? With both sides of single ion channel. You can control, you can study single ion channels. And what are the risks of this? Because you're actually pull it apart, tear it apart. There are risks. Channel property might have changed because severe alteration of the cellular environment. Okay, so I gave you already about the movie YouTube about voltage clamp method. So you watch, if you haven't done, you watch this YouTube, and you are actually looking at a Nobel Prize winning experiments with this. So just briefly go through. So there has been a squid axon, so voltage clamp method was invented by Cole, C. Cole initially, but Hodgkin and Huxley recognized the importance of this technique. They actually adopt to use it to to find out the action potentials contribution by different ion channels. So there's a big, huge squid axon. The diameter is about 1 millimeter. In microscopic scale, it's huge. So you have a saline solution and you put your recording electrode, and of course your reference electrode is outside. And you can measure this membrane voltage. And the cool part of this is now you actually have another electrode, which is a command electrode. And now you can put, because this is a comparator, the different signal can be amplified and you can inject with the current while measuring the current. So what it means is you can set up a command voltage and then you can measure the amount of current. So the current, which is not dependent on the voltage dynamics, can be realized. So here you set up membrane resting voltage, then nothing happens. And when you suddenly change the command voltage into 0, then you see this minus 65 voltage command has to be 0. Then for that voltage, membrane voltage difference, a current has to flow to meet this. So this is a feedback circuit. So it's common and typical for electrical engineers and mechanical engineers or physics students or even chemistry. You know the concept of feedback control. They actually use this to apply. And then what happens if command becomes 0, then now you measure membrane current. Hey, this black line is the result. There's a surge, and then going into this inwards current and outwards current. And this is a remarkable finding. And then they actually use a different blocker for different ion channel, such as potassium and calcium. Okay, and so they actually dissect out different ion channels or properties. Okay, so let's get into more. So voltage clamp and current clamp is a closed electrical system to explore the relationship between the membrane potential and current flow. You can use it in an intracellular or even patch clamp. So the voltage, what I mean is across neuronal membrane, and the current, what I mean is associated ionic current through the channels in the membrane. So voltage clamp means fix, okay? So membrane potential is clamped to a set voltage, as I showed you, command voltage. And measuring current generated by ions moving across the membrane by holding the membrane potential at a set voltage, we call holding potential. There are like different kinds of current you can measure. So what if this is a whole cell, an entire neuron, you can measure the current. That's a, okay, let's see, 한진욱, or an individual or class of ion channels within the neuron. So a single ion channel, the current range is different from the entire neuron. Jinuk, can you guess which one's nanoampere, which one's picoampere? So the first one?
Is nano.
Good, yes. The second, a single,
Is pico.
right. It's easy to say, but picoamp is real tiny. You have to overcome the noise level. So this is a remarkable one by using this voltage clamp. And on the other hand, hey, why clamp only voltage and measure current? The other way around, you set up giving a known constant current is injected while you can measure membrane potential change. That's we call current clamp. So what are we doing with this voltage clamp? Let's say you have identified a single ion channel, which is of your interest, and you want to figure out the electrical properties of this tiny single ion channel or a group of channels, let's say, and that there comes we call by fixing a voltage, and you measure the amount of current, and you change the voltage, minus 80, 60, minus 20, plus 20, you measure the current by using voltage clamp method. And this is one set of the so-called IV curve. Let's say another looks like V and I are linear. Okay, so this IV curve is a curve for each data point of current for a given voltage. So we are talking about a voltage clamp method. And inward current is by convention with negative, outward current is positive. So that's result of positive charged ions crossing membrane from outside to inside. That's inward current. That's a negative here. All right. And so we know, like in general electrical circuits, relationship between the voltage, potential versus current, they are involved a resistance. But here we are more interested in the slope of this. It's x-axis voltage, y-axis current. So it's actually slope is not resistance, but 1 over resistance. And that's called a Dewey. Can you call what that is?
Current resistance.
The answer is not far away.
Conductance.
Exactly. That's called conductance, the slope of IV curve. Okay. So this conductance is really depends on ion channels permeability for which kind of ion, specific ion, and also what concentration of the ion inside and outside of the solution through the channel. Okay. So here is an example that you see the relationship is nonlinear, and that we call, because the current changes depending on what kind of voltage it is. So this is so-called voltage dependent channel. A good example is for our action potential, there is a voltage gated sodium channel, voltage gated potassium channel. Those are this nonlinear shape. Otherwise, a non-voltage dependent channel will not be affected. Only there's just one type of conductance. Okay, so now the question is, how much current flows through a particular ion channel for a particular voltage or environmental condition? So we can study this by studying over time measuring the amount of current. So let me give you a wholesale mode example from the textbook. And let's say we have this bar is about stimulation. We throw an agonist, which is keep making the ion channel to be active, okay? For a ligand-gated ion channel, which means you need a specific ligand, attach it to ion channel that will open the ion channel. So agonist is applied to the bath during this time, and then you measure right after the currents flows, okay? And that's wholesale mode, an example. So the ability of a cell or a channel to pass current under conditions, for example, like compound, in this case, agonist inside the bath, can be shown with current over time plot. Okay, so this is a wholesale mode of patch clamp technique. And I want to also show another one. This is a single channel mode. Now we are talking about patch clamp of a single channel. And, research is always about comparison, right? So before drug and after drug, you look at the differences and before and after. Now we are looking at more of this kind of on and off, like looks like a digital, okay, binary type of signal. So before and after, we see the striking differences. And this is in fact a cell-attached mode of patch clamp. So you are looking at a single ion channel's current, okay, before and after application of a pharmacological agent, which somehow looks like reducing the spiking, not spiking, we are talking about ion channel, looks like reducing the activity of opening of channel. All right, so this, right, is single channel mode, and the thing you want to note is look at the size of the current. It's a picoamp level. While the whole cell, you puncture the cell, you connect, measure the current, is hundreds of nanoamp level. So a huge difference, right? And also, what about the temporal? You have to watch this X scale. This we are talking about seconds. And single ion channel, you are measuring millisecond order. Okay, so rapid and transient events of single channel opening and closing. And this statistical nature is something very important. We understand how neuronal firing and activity is linked to these at the microscopic level. It's related to this even down to single ion channels and different kinds of single channel activity. So for example, just there are disease who has, okay, let me give you a quick example that, hey, there are people who may not feel pain. So in a circus, you see some people maybe fire, they can walk on the fire, and you cut through, pierce, but having no pain. And people realize some of those patients actually have a mutation in that particular ion channel, which is related to a pain sensation. So you actually can figure out what's wrong with this patient. Of course, this is a little genetic. I'm not sure how to treat them, but at least you know the mechanism, then you can come up with how to resolve this issue. All right, now we change our gears into like the real sample preparation. So first, in vitro, there's a demo for primary culture. And in vitro experiment, you can isolate the cell from a region of interest in the nervous system. Let's say I'm interested in cortical neurons, you plug out the cortex. And I'm interested in trigeminal neurons, then you have to go to trigeminal and then plug them out, okay? And isolate. So that's called in vitro. I'm not talking about cell line, which you may be familiar with a cancer biology. People use cell line, which is immortalized, but this is a primary. Primary cells are not immortal, so you have only limited time window to study them. So what's good about this is reveal properties close to those found in vivo. Hey, it's not in vivo, but we are trying to mimic while still providing. Then why are we trying to do in vitro experiment? Because in vivo, you cannot see what's happening inside very easily. You take out, you can have an access, excellent access, and you can control these individual channels by putting drugs into the bath, changing the environment and directly measure. So that's a great advantage and for you to study. So for example, is knowing neuronal physiology when the extracellular concentration of particular ion or neuropeptide increasing or decreasing. So neurophysiology can be well studied. Okay, I want to really show how things actually happening. So it's best to do it your hand, but now I'm just showing a picture and photo. So let's say I'm interested in studying a slice cut. So one is primary cell, but the limit is cell itself, everything is microenvironment and connected. So there's a limit. So in between trying to mimic in vivo and in vitro cell, there's a limit, an alternative. You just make a fresh brain slice like hamburger steak and make them keep alive and you study them so that you keep some of the microenvironment, okay? So how do we do that? Hey, there's a mouse or rat and you can very quickly cut the neck off, okay? And then you take off the brain with a spatula and you know that you are interested in, let's say, some part of hippocampus for studying memory and learning. Then you cut it off and you see, look at that. And then you use a vibratome to make a steak slice. And you see this, but the slice has to survive. So you have to have a solution called artificial cerebrospinal fluid. And you have to keep bubbling the oxygen too, because you don't have now blood vessel. And this is very tough. And critical part. And then you can put it into your electrophysiology rig, and you can do study with a patch clamping. And you see here, this is an interesting one that you can make this, these slides can be like flowing because you have to keep flushing media across, okay? So you have a, female nylon stocking. Initially, people still use it to attach to this clip and then use this one to fix the brain slice. And you see these nylon stockings, nylons, but here you put electrode. You see, look at these two electrodes, and you can start study this with the electrophysiology. So what's the advantage of brain slice? Easier to make. Now we are talking about extracellular recording without periodic pulsing of blood, because this microscopic scale, pulsation or blood can actually interfere, okay? So we study from a particular region of brain. Hey, I want to study hippocampus, which is inside the brain, and it's very hard to do imaging directly because there's a cortex outside. So a good way is cut and expose that region of interest directly so that you can have internal brain structures. You can actually study them directly. And because it's exposed, you can actually pour pharmacology, drug directly. So you can study the pharmacology of known synapses. And studying specific roles of individual neurons, the neurons are there, but let's say you want to study memory or neuronal plasticity involved in learning and memory. So the memory you can actually study with this, let's say, exposed hippocampal slice we call. The interesting part is, in this case, that neuronal connections somehow has to be within the slice, very thin slice. So then you can study this. So study neuron in circuitry. Okay, so that's a very important aspect. So brain slice is placed in a chamber that's continuously bathed in a solution containing proper proportion of inorganic ions, neurons, and gases to allow neuron to survive. Okay, so in fact, the electrophysiologists say to me that 99% success is based on how you prepare the slide. So a novice can make the slides to survive for 3 hours. So she and he may have only limited time. A professor who have decades of experience can survive this 9 to 12 hours. So you can have more time to do many experiments. So here's the example showing that reservoir, and you have a pump, peristaltic pump, to push this media, and bubbles are harmful for the slice. So cells do not like to be touched the bubble. So you have to trap the bubble and microfluidic chamber. You flush the brain slice. And then while you have an electrode to study, you can touch with a patch clamp or extracellular recording. And this is how things work. So brain slice preserves some endogenous connections. You cut it off, okay? So there's a limit, but luckily you can have some still connected, not like in vitro primary cell culture, okay? While still providing level of access. The good thing is you have access available through this cell culture condition. So you are trying to closely mimic in vivo environment. And as I say, this is kind of artificial. I mean, cut the slice, this is still quite acute injury type. So usually people study on the same day. We call it as acute preparation. Of course, some people do more chronic preparation. And when you cut the slice, think about the circulation. Blood vessel has to be around to provide nutrients and oxygen. And now you have to be solely dependent on diffusion and convectional transport of oxygen and nutrient. So the slice cannot be thick. So typically it's a 200 to 500 micron, typically 350 or 400 micron. If it's too thick, the inside will die out, okay? And when you cut, the nearby cut 50 microns, neurons are damaged. So this is a kind of, you need to know. How do we use slice? Using a tool called Vibra cut is tome, okay? We call it Vibra tome. What do we mean by Vibra? Your knife is vibrating and to cut, to make a slice, okay? So that I showed you here, that we have a holder and sample while it's vibrating and moving. So you have a brain cut and then come back, you push 400 micron, you cut back 400 micron cut, so that you can make many sections to study. And the 3D shape looks like this. And this vibratome is in our laboratory. So when you have a chance next time demo, take a look. This is expensive equipment, like actually $17,000. It's pretty expensive. So still, many neurons remain healthy despite mechanical shock and damage of slicing. So that's how we use it for slice study. So in vitro, the neural specimen has to be incubated in a well-regulated bath solution with a 95% like oxygen, okay? The composition of the solution is critical. Neuronal electrical property depends on the concentration gradient of ions in and outside of the cell. So one of the questions of, hey, what's the action potential, resting potential difference? In fact, I heard electrophysiologists who actually did in vivo recording say, hey, resting potential is not the same as what we memorize, 60 minus 60 minus 7. It can be minus 40 millivolt, something like in vivo environment, there are whole different things are changing. So the composition of solutions is important to make this study. And patch clamp, the solution inside the microelectrode should mimic intracellular milieu, typically composed of salt. You know, we have to mimic, maybe not complete, but as much of the inside the cell cytosol. So salt, ionic buffering agent, and a pH buffer. So better to see how it looks like, okay? Setup, electrophysiology. I just want to show you that there is a perfusion inlet and outlet output, and you see brain slice, and you have to have a measuring electrode, and also you have to have a reference electrode, okay? And this, you see these ones are not easy to see. It's a microscope objective. You are looking from the top to bottom, and you see this electrode, you can identify cells of your interest. So you touch the cell on the surface, you cannot see ion channel, but you can actually see it pretty closely. And by measuring this electrode, you can see whether you have a good seal, you can study. And this right side one is actually, we've done this study, published in Scientific Report 2 years ago. So I just want to show an example that, hey, there's a brain slice and electrode. This is a hippocampal slice. This is a cartoon called CA3, CA1. This is studying so-called long-term potentiation, synaptic connection changes with respect to, you see coil. So what our question was, hey, magnetic stimulation will be affecting the memory. What's the mechanism? So that's our in vitro study. So we made a magnetic coil. We give a magnetic field to this, and we measure the electrophysiology, EPSP, so long-term potentiation and relevant ones. So this is just one example in a research setting. So what's the advantage of in vitro electrophysiology tissue prep? Hey, it's easy for physical visual access to individual cells. You know, once you get inside the brain in vivo, it's very hard to see and know. So it allows detailed studies of the molecules and proteins that affect neuronal physiology. So for neurophysiology, this in vitro study is very much critical, because you have a good control over cellular environment by using culture or bathing media. to finely dissect cellular, subcellular, physiological events. Okay, for example, you can block this receptors at the synapse by specific blocker, so that in the end you can find out the role of this AMPA receptor and NMDA receptor. This sounds a little weird, but this is so famous. The most abundant receptors are AMPA receptors, and NMDA as a neurotransmitter. This receptor as a ligand is NMDA. So this is used for the mechanism of synaptic changes when you study neuronal plasticity and memory and those. Okay, so I already discussed this about heterologous expression system. I think I already covered this before, so I'm gonna go a little quick. But the point is we can express mutant form of a channel, and that effect of molecular changes on a channel's electrical properties can be studied. So this heterologous system is using a kind of a different type of cells, such as CHO cell and HEX cells. And these cells are easy to culture, and we are studying some specific channel. Then these cells do not have many inherent channels itself. That's a good thing. So that's few endogenous channels. So it's clean cells, and they take up and express a foreign DNA. Let's say I'm interested in specific channel, and we clone this, and we insert this channel into the cell express, and making this channel express on this specific cells or oocyte, xenophos oocyte, then we can do electrophysiology for this. Okay, so this is an example, xenophos oocyte, and you can have a cDNA, complementary DNA, inject, micro-inject, and express those specific channels on the surface. You can do voltage clamp with this huge egg cell, and you can do patch clamp for this. And this is a little detail, so that you can study with this model system, study specific ion channels. Okay, in vivo study, so far I talked about in vitro recording, but it may not reflect accurately the neural activity in intact organism, which is ultimately our interest. And so, but it's very difficult to do in vivo recording. So most in vivo recordings will be intra or extracellular. Of course, it's extracellular because you cannot see it well, okay? Difficult to visualize, difficult to precisely position, and you use electrodes, tetrodes, or multi-electrode arrays. While, at the forefront, some people can claim that, hey, I've done in vivo intracellular recording with a very sharp pipette to impale you through the cell. So yeah, those are, one can report, but it's very, very difficult. So in vivo, there are two kinds of recordings. One's acute versus chronic. So acute means you do this experiment and you kill the animal, okay? It's done one time. Chronic is over and over. So acute is over hours and chronic over multiple days. And so you use animal once for acute recording. If it can be too damaging to the animal, so you may not keep it. Okay, chronic so needs a cannula or surgical implantation of electrodes for long-term access to recording site. So even multi-electrode arrays you can actually implant, so to monitor neural circuit activity during complex behavioral tasks over long periods of time, or during a variety of different conditions and multiple tasks. So in that case, you do need a chronic recording. And sometimes I already showed you C-level chambers implanted on a skull to placing new electrodes within the brain over periods of months in rodents, for example, like windows and electrodes, or non-human primate monkeys we can study over years. So I think I'm gonna go anesthetized versus awake. And I just point out that we mostly do anesthetize animal, which is great. The animal doesn't move. However, we are affecting the system. Excitability and neurotransmission will not be the same. So the limit is we are actually artificial or controlled and for paralyzed animals. And we have to eliminate breathing motion for reducing noise. So all these ones actually as limitation. It's a good thing, but a limitation. So ultimately, you can see all the fields is going, what's happening in a awake animal. Of course, this is more difficult, okay? Why? Movement artifact and signal noise. However, awake animals is the more ultimate goal. So that we can correlate behavior to the neural activity of individual cells. You know, this is fascinating. Hey, I'm doing this behavior, let's say fear behavior. Can I identify fear neuron? That's amazing. I recognize the face of different, people. Let's say we identify neurons who recognize the face. So you can possibly correlate. And this single or multi-unit recording during this behavioral task to examine neural basis of various cognitive and behavioral phenomena. So I'm not saying it's easy, but that's the difference, and people are going towards more of awake. And so this is the last one for manipulating neural activity during electrophysiology experiment. So let's say we are considering loss of function, gain of function. We are interested in certain neurons during neurophysiological recording, and these neurons you can actually inhibit or activate. And how does this relate to our question? So let's say a hypothesis. I'm curious about this neuron A and neuron B. My hypothesis is neuron A will stimulate neuron B. Okay, so how to test? Then you have to make neuron A to activate. So you need to stimulate neuron A first, and then during the time you are monitoring the neuron B activity so that you can test this relationship between the neurons. And another example is contribution of certain type of neurons to a physiological process. This is an example that, hey, there's a so-called GABAergic neuron or cholinergic neuron. For example, GABAergic neurons, and this GABA is a chief inhibitory neurotransmitter. Okay, so neurons that produce GABA is called GABAergic neuron type, and you can connect this one to a physiological process. So for example, you can inhibit only these neurons while examining physiological property of a neuron of interest. So by inhibiting or stimulating, you can study these specific type of neurons and related to electrophysiological behaviors. Okay, we've gone through quite a lot. I hope you can review well, and I'll give you now